Selamat pagi. Good morning, all participants. Thank you for joining this session. So this second half of the breakfast talk is on drug delivery, and proper drug delivery is very crucial for clinical outcomes and therapeutic outcomes. So therefore, today's topic is on drug delivery, a topic which is microneedles unlocked for faster, safer and smarter healthcare solutions. So the speaker for today is Dr. Mohammed Jasim Uddin, whose expertise is in this field. And allow me to introduce Dr. Jasim. So he obtained his PhD from Greenwich University, UK, in pharmaceutical sciences. He has more than 14 years of research experience in drug delivery as well as in pharmaceutical sciences. His more the core research area is on microneedle technology with transdermal drug delivery systems for both small active molecules as well as macromolecules, using techniques such as 3D printing technology as well as molding techniques. And besides that, he also worked with formulation scientists as a formulation scientist in Pfizer, UK. So therefore, he is also aware of the industry requirements for drug delivery as well as advanced drug delivery systems. Besides that, he also has a lot of collaborations in this field with other universities such as the University of Kent, University of Belfast, University of Strathclyde, and of course his parent university, that is Greenwich University. And all the researchers are in touch with him, and the collaboration is resulted in high impact publications, not only for him, for as well. So he also proud recipients of many awards. A few of them are Young Research Scholar Award in 2022 from World Academy of Sciences, Young Promising Researcher Award 2020, Young Innovator Award 2020, both scientific communities across the world as well as in Malaysia. So he's also active member as researcher in reviewing the journals as well as advisor to some of the publishers, Taylor and Francis, Springer, Nature, MDPI, as well as a guest editor for reputed journals. So we have here Dr. Jasim. Thank you for your time and accepting this topic. So without any further delay, I will pass on to Dr. Jasim.
Okay, thank you very much. Am I audible?
Yeah, audible. Go ahead.
Okay, thank you very much, Dr. Shaik, for your brief introduction. And thank you for inviting me to the breakfast at UMHealth. So let me share my screen first, then I'll be proceeding to my knowledge sharing and presentation. Okay, good morning, everyone. As you know, my name is Dr. MD Jasim Uddin. I'll be today discussing on a very innovative topic, which is quite new topic for Malaysia, especially on 3D printing and also the microneedle drug delivery system. And it might come up with a potential application in the field of medicine or in the field of drug delivery. Okay, so those who are not familiar with me or might not be able to connect with me because I'm serving for almost 3 years now. So this is I just wanted to give a glimpse of my career, what I did actually. I think from here, we can come up with some good opportunity to collaborate and connect, right? So 14 years of experience, obviously, I'm a bit blended with both academia and industry experiences. Also, I've achieved higher education, teaching and learning from certificate from Harvard University of Teaching. I actively organize almost more than 10 international and national conferences or events, attended more than 20 professional trainings, active member of 5 professional bodies, including Malaysia. So I've achieved 35, almost 35 external recognition and 10 awards. The most interesting thing for my career is I have achieved around more than 10 research grants, including the international and national grant, which is worth almost $1 million. And currently, I'm working on a book which might be the contributions in the science, I can say, after 14 years of the experience. And this book will be coming from Spinge and Nature on specialized on the drug delivery system and microneedle. And I publish a few articles, and also I supervise almost eight PhD students currently, and also masters six students in my lab actually. So this topic might not be the same topic, that this is a blended topic of engineering, drug delivery, clinical applications, right? So before moving forward to the actual topic, I would like to give you a sort of idea about 3D printing. I know 3D printing is quite familiar to the society because we are going to print a lot of toys, or might be the kids nowadays are using 3D printing to fabricate or print different type of structures, especially the engineering people. So what was my intention? My intention is to take this opportunity to transfer this knowledge and also technology from engineering to the pharmacy or pharmaceutical sciences. So 3D printing of microneedle could be one of the potential areas that can be explored actually. The reason why I decided to move to 3D printing for the printing of the microneedle. And those who are not familiar with the microneedle is something which is a bit tiny structure, and this can be the alternative for the injections for the delivery of the medications underneath the skin. It is a bit short. It will not produce any sort of pain underneath the skin, and just releasing is drug underneath the just upper portion of the dermis. So, if I would like to take the opportunity to print microneedle with the 3D printing, it will be allowing me to give a easy and fast personalization and customization, especially. And also we can tailor the dose. As for example, at PPUM or any other hospital, if you see in Malaysia, we are giving the medication, especially for the chronic diseases like cancer or any other patient. So we are not actually trying to give a dose which is personalized. Actually, personalized is there because the dose is coming from the company. But what would happen if we can actually set up the whole setup for the personalized medication, especially for the people who are suffering a lot. So like cancer drugs and other, they are quite sensitive, right? So we can give or print the medication in the hospital in as a precision medicine and give them as a dose, right? So this could be one of the interesting opportunity for us to deliver the medication. So actually, what I actually wanted to improve previously, the microneedle was printed through the micromolding technique, or you can say fabricated. But now we are directly printing them through the printer, so we can reduce the step which is required for microneedle printing. So we do not need two steps now; we need only one steps. So now you can see in this picture there is a as a video, you can see we are using the stereolithography uh, because in 3D printing there are few methods that we can use. So stereolithography is something which will help us to fabricate a tiny structure layer wise polymerization of photosensitive polymer through the curing process name called photopolymerization. So in our study, what did we do? We have synthesized our own resin in the laboratory. The resin that is available outside is quite expensive, and it can affect the overall healthcare cost. But this one, class 1 biocompatible resin, can be synthesized in-house, so it can reduce the healthcare cost as well. The printer that I use is Formlab from the Formlabs company. So if you see, everyone's research is quite motivated from something, from might be personal experience or might be from the society for the country. So my first motivation was my mother actually. My mother was a diabetic patient, is a diabetic patient. So I have seen her every day picking up the blood for the measurement of glucose, and all the time she is just giving the insulin 2 times. So I was a fit feeling very sad because my mother has to take 2 times of the insulin dose, right? Not only my mother, but also the people around the world. So many mothers are there, fathers are there, even the small kids are there. 422 million people are worldwide suffering as per WHO. So first motivation was I wanted to do something for the diabetic or the people who are suffering with diabetic. So in here, what did I do? I tried to print the microneedle. First of all, I designed the microneedle by using a software called SolidWorks, and the patch that I made is 15 by 15 1 millimeter length of the needle. Okay, so the minimum that we can print by using 3D printing is 1 millimeter. So we put 15 in one side, 15 in one side. So total, if you can consider how many numbers of needle we can print. And then I printed two shape. One is pyramid shape. The structure has been taken through SEM, scanning electron microscope. You can see this is a bit look like as a pyramid. So we put the name as a pyramid shape. The other one is a sphere shape. Okay, so the array was printed by the SLA printer, and once I print them with the SLA printer, I just wash them with the isopropyl alcohol in order to remove all the unwanted material and cure them 60 minutes for 40 degrees Celsius under the UV radiation. Curing is important because if there is any unwanted material is producing during the 3D printing process, it can get removed by using this curing process. It will, because of there is a synthesis process. So ultimately, the process might come up with some unacceptable material. So we can remove them by using UV radiations. So then what did I do? I make my own formulation. The formulation, it is quite tricky for us to load the droplets or load the insulin on the surface of the microneedle because this is the tiniest structure, and in the naked eyes we cannot just see them around, right? So what I did, we wanted to produce insulin sugar film, especially which will be coated onto the surface of the needle. And in here I managed to formulate three formulation. One is insulin xylitol, insulin mannitol, insulin and trehalose. The reason why I put insulin with the mannitol, xylitol, because of this will help to maintain the structure of the insulin, because insulin is a bit higher molecular weight per material. We need to maintain the structure. If the structure is just dictating, we cannot just get an optimum effect. So what did we do? There is a patented process called inject printing. So the droplet of formulation, we just put them into the tennis spots onto the needle and we coat them. So this one is a US patent from our group. So this is a patent from my PhD thesis. So this is a group that actually I worked with during my PhD. So if you see this one, so this is a pattern by which we can coat the microneedle onto the uh, we can coat the material onto the surface of microneedle. If I can see, if you can see this video, uh, you can see there is a small nozzle coming onto the microneedle and just dropping, or you can say jetting 5 dot onto the surface of the needle very precisely and accurately. This will help us to reduce the material lost because the materials are quite expensive. So every microneedle is getting almost 5 droplet. Okay, after some time it will be dried onto the surface of the needle. So this is one of the microneedle, stainless steel microneedle, obviously FDA approved. So we took this opportunity to print them, and this has been actually already patented and is industrial use. And in the picture you can see we managed to put the drop onto the surface of the microneedle. So there are some pictures that I do not want to ruin your days because of this is a complicated picture, but I just wanted to show actually how we did the micro CT image to see around how we load the medications onto the surface of the microneedle. So these are all cross-sectional study that can be done to our study. But this is one of the interesting study that I would like to highlight. So what did we do? Initially, we have run through one experiment for doing the in vivo or testing onto the mice. So in here, we actually introduce the diabetes. How we introduce the diabetes? We just inject them with the streptozotocin, which is a diabetes inducer. So we observe them from day one and day two. After 7 days, what we realize, there is a development of the diabetes into the mice. So then we start to realize to treat the mice to see the anti-diabetic effect. So we have run the experiment for 4 hours, and in the right hand side, if you see two graph is there. So we had taken three groups. One is untreated group, which has not been touched on the negative control. We also already compare with the marketed available injection, subcutaneous injection, the same dose of zero point 0.2 IU, and also we have also load the similar material in our 3D printed microneedle. So what we have seen actually, if you compare between two results, like green one is representing the subcutaneous injection from the market, and the blue one is representing our microneedle. So after 1 hour, it was almost following the similar pattern, right? Because we have given the dose subcutaneously. But what happened after 2 hours? The suddenly it maintains the plasma global steady state, but for the subcutaneous is actually reducing its impact because it is just directly given everything into the subcutaneous region, and slowly it will be just coming up additional no effect on that. So later on, because to confirm this result, we did take some blood for the plasma insulin concentration measurement. So what we actually realized, our microneedle was giving a maintain or control release of the medications within the body, which actually helped to maintain the blood glucose level actually for a longer period of time. So we get the opportunity to deliver them, and ultimately we realize that we can actually use this microneedle for the drug delivery of insulin. And later on, we got a gift, obviously. So this paper has actually published impact almost 7.34 Q1 paper. We got the similar study, which has been this study has been published actually so far by our research team. So then, what happened actually? My life was going very well, but suddenly I have heard about my one of my undergraduate project supervisor has been diagnosed with breast cancer and other type of cancer, I can say. So then she died. So I start to realize, can I move my research to the cancer treatment? So then I start to realize to do my PhD with the cancer, honestly speaking. And that if you see 9.6 million people are worldwide are suffering with the cancer, which is not a less number, is a big number. So what did we do? We designed with the new microneedle, it's called cross-shape microneedle. And here we can see the scanning electron microscopic image, which is an image that shows that this is a cross shape, and the colorful things like the red color shows that the loading of the material, and the other picture is there in the right hand side of yours. This is the 3D picture. Actually, you need to wear a 3D glass to see the real information, so unfortunately we cannot observe it now. So how we test the penetration of this microneedle? We test the penetration by using an optical coherence tomography. Optical coherence tomography is a technique by which we can see up to 2 millimeter of the penetration of any material or components. So in here, what did we see? We compare actually in between the results, right? So we have seen the microneedle penetrated very well. If I consider more than 90% penetrations are there into the skin, because of sometimes we do not believe that the microneedle can penetrate the skin. And if I see the picture, I see here that microneedle penetrated just upper portion of the dermis. And in here, in the right hand side of yours, if you see the picture, when we deliver the medication into underneath the skin by using microneedle, after 4 hours, the material has already been dissolved within the skin by passive diffusion. So it confirms that microneedle can actually deliver material within the skin. So this is the interesting result that we actually found so far. We tried to develop the skin tumor into the mice, and in here we got four groups: control animal with the tumor site treatment and non-tumor site treatment and change in tumor site treatment. So if you see the picture, after some time there is a development of tumor, but the tumor has been reduced when we change the treatment site. Means if we are penetrating the microneedle beside the tumor site, there is a huge opportunity that we can get rid of the tumor. And the tumor reductions is within the 3 days without negotiating the life of the mice after some time. So if you see this picture, we have come up with we managed to get some histology as well. So I'm not expert on this, but still there are some collaborators who help me to define. But within the study, what actually we got, if you see this one, the change in treatment, non-tumor site treatment will be giving the best effect within 4 days, eradicating the cancer or tumor from the site. So because we just need to give beside the tumor, not onto the tumor, right? So because it is damaged. So we got the findings and decided to submit the paper. So we got another gift from Material Science and Engineering saying the same 7.34 impact factor Q1 paper. This is the paper that has been submitted as a part of my, I did this study for my PhD. It took around 8 years to confirm the outcome of this experiment. Then I realize we are giving the medication to the patient, but it is not controlled. How can we control them? This is also important part because sometimes if I'm giving the dose right in a solid form, sometimes they might not be getting the optimum effect. So that is the reason. What did I do? I tried to come up with a concept when actually I wanted to deliver the medication in the liquid form. Previously, the microneedle was developed to deliver the material in the solid form because of solid material will be dissolved and also by passive diffusion it is going into the skin. But after some times, I can see there might be some clogging or might be not enough interstitial fluid which can dissolve the material. But within this technique, what did we do? With 3D printed microneedle, then we develop one micro electromechanical system. What is the micro electromechanical system? This will help us to control the medication. If I want a small amount, a small volume from picoliter to nanoliter, we can control them. This will help us to give the personalized medication. So, but initially, the our life was not easy because we tried to we tried to make a hole into the small tiny needle. It's not easy job. It was very tough job for us to make a small hole. So pebble type things we have designed and we try to print them. Unfortunately it didn't happen. Ellipsis one we also tried them in the surface, small needle, small surface, small hole we wanted to print, but it's not happen. But ellipsis 2 it happens. How it happens, actually I want to show you the real picture. Once we tried the bevel type picture, you can see there is no formations of the nozzle. But ellipsis one, we found some nozzles formation, but unfortunately the nozzle was not sustained. But ellipsis two, we were successful to put a hole in the side wall of the microneedle. Then what will happen ultimately? Because that is when we put the hole into the side, because when we penetrate into the skin, there is a pressure that is coming also from the skin, which can block the nozzle, because this is a very tiny nozzle I can see onto the surface of the microneedle. So in there we design one customized microdosing pump, and ultimately which can load up to 5 micrometer. This is one of the indication for the technology transfer. So later on, if it is going for commercialization, our 3D printed microneedle can be transferable to the companies for the commercialization to deliver a wide range of medication. This can be also customized, and also we can put them together with the microneedle device. So what did we do? We tried to deliver the liquid insulin at this time, and also we ran the experiment for the first time for 6 hours. So ultimately, what it happens, ultimately we see there is a steady state plus glucose level maintenance after 6 hours. So it will help us to see whether this 3D printed microneedle MEM system is sustainable for the delivering of insulin within the mice, right? So this is the thing that we wanted to do the same. So we got another gift after some times in 2021. We published our paper in impact like 11 point I think T9 Q1 paper in additive manufacturing with our team. I was also a co second author. Ultimately, this paper was going with the good citations as well. So after that, what happened? I decided to move a bit forward with the osteoporosis. Osteoporosis is also because I never delivered any drug for osteoporosis. My motivation was somewhere because 8.9 million people are doing fractures annually every 3 seconds. Even by 2015, if you see, there is in the man is projected to increase almost 310% of osteoporosis fracture, right? 240 in the woman. So if I see, denosumab is one of the widely used drug, which is a monoclonal antibody. The molecular weight is one of the challenging part, 147 kilodalton, which can be used to deliver through the microneedle. So what did we do? We saw minimal. I'm so sorry for the picture. I had to be a bit, obviously it is under the ethical approval. So I had to, do the my in vitro studies by using the mouse skin, and also this is the important part. So we did not only check the amount of osteoporosis, but also we check the the metal ion that is related to the osteoporosis as well, the bone formations or other thing, iron, copper, and also magnesium and zinc. So what did we do? We first of all took a healthy rats or mice. We developed the osteoporosis after bone deformation after 4 weeks of operectomy for the female mice, and then after 6 months of treatment, this is the first time we did the uninterrupted study. Three times we had the failure of the study, but fourth we got the results and we managed to survive actually the mice for 6 months. And this is one of the miracle happens, I believe. So after 6 months, what we see there is a development of the osteoporosis condition, and also we could maintain the uh, ion related to the osteoporosis as well. And after that, recently we published this paper as well, 2024, in the Molecular Pharmaceutics from American Chemical Society. I was leading this work. So in here we have designed this microneedle, 3D printed microneedle injection system. This can be also used. So now what we are thinking, we are trying to remove injection from the market, and we just replace this microneedle with the injections. Sorry, injections with the microneedle, right? So that can be used as a to improve the patient compliance. So this is it. I think this is it. That is all about my things actually. Thank you very much.
Yeah, thank you, Dr. Jasim, for concising all the important aspects of your research in such a short time. Very impressive and excellent talk. If you have any questions, please post your questions in the chat box. We can take a couple of questions. Due to time constraint, we can take a few questions. Meanwhile, Dr. Jasim, before we get any questions from the participants, what about the cost effectiveness of these microneedles? Because scaling up from the point of industrial scale, um, this technology, because you are also work with the industry, so how good is the scaling up of these advanced techniques?
Okay. So if I consider the 3D printing process, right? 3D printing process previously can be printed by buying some, resin from outside, like from the company. It will obviously, in the research level, it will be increasing the healthcare cost. But when we are going for commercialization, we'll be thinking of the healthcare cost as well. So what would happen once we have actually developed the resin in house by using our own mechanism. So we could reduce the one-fourth healthcare cost. And if you see the medication, the microneedle is not used to deliver the paracetamol which requires bullet dose. We need to have the targeted dose. So I am actually targeting some cancer patient, diabetic patient, osteoporosis patient, and also some other chronic diseases listed by WHO. So in here, a small dose variation can actually threat the life of the patient.
Yeah.
So if I see recently, I would like to, though this is not relevant, but still I would like to highlight. Recently, one of our innovation has been showed into the Putrajaya festival. My lab, my member has actually developed one cardiovascular stent. And when I start to realize the healthcare cost, it can reduce the one-fourth cost of the healthcare, because in Malaysia, one stent costs around 10,000 ringgit. So my one can be delivered with 2,500. So if you see the cost effectiveness, once we can do anything in-house and
Yeah.
mass production,
Yeah.
I
Okay.
think
Okay.
overall healthcare cost will be decreased.
Okay. So this is basically for transdermal drug delivery. Can it be used for other routes of drug delivery as well?
Of course, it is
Yeah.
because of we have already made for implants, we have made for cardiovascular stent. So unless we can made it up with the biocompatible and biodegradable resin, we can
Mm.
use it for so many applications. But the application has been started because FDA just approved the delivery system as a 3D printed product from 2019. So people are a bit concentrating on this research.
I'm so sorry I have to end here because we are running out of time. So thank you, Dr. Jasim, for your excellent talk as well as giving insights into this topic. I hope this might have inspired many of the researchers in UM as well as beyond UM. Please feel free to contact Dr. Jasim. He is in-house in our faculty. So this is one of our niche areas of our faculty as well as in our department. So feel free to contact him, and he's having in this field worldwide network. as well as with working with international experts in the microneedle technology as well as 3D printing. So with that, I will conclude and end this sessions. Thank you very much for all those who joined this session for participating, and have a nice day. Thanks.
Okay, thank you very much. Thank you. Take care of yourself. Goodbye.